Thank you, Brenda. And now, before we go any further, I want to express my personal thanks to my two very distinguished colleagues who help us to put on this incredible conference just for you. First, I want to really thank Brenda Stoddard. Brenda, you were the force that brought us all together way back in 2012 with a new idea to partner together with a joint conference. And look where we are now, all these years later, 13 years. And second, I want to thank Larissa. When Brenda reached out a few years ago to you and your team to expand the program to add a third track that truly made us whole, you and your team's response was very simple. Absolutely, let's do it. What do you need from us? So Larissa and Lonnie and Stacy, I really want to thank you for just making this a great conference. This is our 4th year in a row with CBER. And so I want to give my colleagues a nice round of applause for them. So for everyone in our audience online, welcome, and in person, I have a question for each of you. What were you doing 5 years ago? Do you recall where you were? Now, if we're being really literal, 5 years ago to the day was May 29th, 2019. And some of you might have raised your hands to say, I was in Boston. And if you were, I thank you, because that means you were at REdI 2019 with us in person. That was the last time we were together, in this hybrid format, in person and online. So if you were with us in Boston in 2019, and now 5 years later, you're with us here, I thank you for your continued participation and engagement as we put on this conference just for you. Now, for many of us, 5 years goes by very fast in general. And if we think about it, so much has happened in our lives during these last 5 years. We've endured a global pandemic, and many of us in the regulatory business learned to say the acronym EUA a lot, especially in our world here. In the workplace, we learned new communication platforms like Zoom and MS Teams. Definitely, those have become part of our parlance. And at home, many of us took to heart the spirit of online shopping and delivery for just about anything that you can imagine. The one constant through this all is change. The world continues to change and move forward. And when it comes to technology, this is no more apparent than the explosion of artificial intelligence that has made ChatGPT a household name. Now, one of the more interesting statistics about the FDA is that we regulate 20 cents of every gross domestic product, or GDP, dollar in the United States. Now, that's 1/5 of the US economy. An incredible stat that really illustrates the vast impact of the FDA. And not surprisingly, the FDA is at the epicenter of where change meets growth, of where medical innovation occurs, and with the ultimate aspirational goal of bringing medical products of all kinds to patients in need. So it's only fitting that this theme forms the topic of this year's conference and this year's plenary session, that is that of innovation, and specifically innovation in medical product development. And to bring this forward-looking topic to you today, we have 3 very special guests for you, which I'd like to invite to join me on the stage for today. Dr. Patrizia Cavazzoni, Director of the Center for Drug Evaluation and Research, or CDER. CDER regulates a whole range of drug products, including those that are over-the-counter, prescription, or generic, as well as biological therapeutics. And a fun fact, now that we're heading into the summer season, especially with me with my head, this includes greens. Dr. Peter Marks, Director of the Center for Biologics Evaluation and Research, or CBER. CBER regulates various biological products, including blood, cellular, and gene therapies, and of course, vaccines. Products that are derived from living sources. And Dr. Jeff Shuren, Director of the Center for Devices and Radiological Health, CDRH. CDRH regulates a wide range of medical devices and, interestingly, electronic products that are not medical devices, but which emit radiation, such as TVs and microwaves. That's the RH part of our acronym. Please join me in welcoming our esteemed faculty for the plenary session titled Innovation in Medical Product Development. So for those of you familiar with our plenary session, you might recall that we historically start with a presentation by our faculty and then follow this up with a question and answer segment with you. Well, in the spirit of innovation, we're changing things up even for our plenary session. We're going to make this very interactive with you. We'd like to really engage with you for this session. So we will not have an initial presentation. We will immediately begin a roundtable discussion with our esteemed faculty here, covering a variety of topics on the theme of innovation. Now, this is where you and our audience, both online and in person, come in. So first, let me speak to those of you who are online. Welcome again for joining us. Please send us your questions online. You can do this by using the QR code that you should be able to find on your screen. Click the code, which should take you to our Q&A website. From there, you should be able to select the session Day 1 Plenary, see the names of our esteemed faculty here, and then please type in your question. Larissa out in the back will be receiving your questions and sharing them during our program. Now, for those of you who are joining us in person, let me speak to you for a second. As you can see, we have some microphones that are set up throughout this room. So you're not standing for our full hour segment. I'll give you a heads up when we're ready to start receiving some questions. And when we do so, please, I welcome you to come in and join us and share your questions. Now, speaking of the question, I do have just a couple of asks. Number one, if you'd like to direct your question to one of our center directors, please just let us know if it's specific to devices or drugs or biologics. You are invited to ask a broad question and invite all of our faculty if it's a broader question there. And then the second note, I appreciate that this is a very priceless opportunity to engage with our center leadership, FDA leadership. I do ask that you focus your questions on the theme of innovation, on the topics that we're covering today, and that they may have already discussed. So I appreciate that. So with that, let's get started with our session. Thank you again for joining us, and we'll get going. Our name tags. Okay, we'll put it here. Well, thank you for joining us. So as we did a little bit of an intro with our center directors, I'd like to kind of continue the introductions a little bit. So for Peter, I'm going to start with you for this first question. We'd like to get to know you a little bit more. So prior to you coming to the FDA and in your current role as the center director for CBER, can you share what role you might have had in your prior life that involved innovation?
Yeah, so prior life, which prior life? I think when I worked in industry, I actually did work on drug development in the oncology area, and for a time we were working on a targeted therapy that was basically targeted for a very large indication. And in generally, innovation for me has come by necessity. Necessity is mother. That saying that necessity is the mother of invention. We had a very large indication that we were trying to use this targeted therapy in, and the trials were going to take a very long time, and it was a very competitive field. And so the question was, how could you speed things up, get to market, and not be last to market, but instead be first to market? And so the innovative approach was rather than using a very, very large indication, to go for a very, very small indication where there was a tremendous unmet medical need. There was not any other therapy in that particular area that was effective. And it took what was going to be a 5-year development program and brought it down to about 1.5 years to 2-year development program. That was done out of kind of desperation because otherwise I think we would have just not gotten there. It turned out that the large indication failed when someone else did the trial. That innovative approach, which was successful, was a reasonable way to go. But I think that type of use of innovation out of necessity that has also paid off during the COVID pandemic and at other times as well.
Excellent, thank you. So you've been in the shoes of our audience with being on the other side of the FDA.
Correct.
Here you are now. Jeff, I want to ask you the same question. Any thoughts on your prior life prior to coming to FDA?
Well, before coming to FDA, I was in academia. So I was a user of the innovations, but I also saw the impact when needed innovations were not available, what that did for patients. And then so coming to then the FDA, I came back to CDRH 15 years ago. And at that time, developers of innovative technologies did not bring those innovations to the US first or second or third, sometimes not at all. They went to other countries. And that was in part due to us at the FDA. And credit to my team, we did a lot of soul searching and analyses on why that was the case, and we made a commitment to change. And now, fast forward 15 years later, more than 50% of developers of innovative technologies for US market do now bring it here first or in parallel with other countries. And we did it three big things I'll highlight. One, a culture change for us, built on the pillars of safety and innovation. We saw it as part of our mandate to advance responsible innovation, innovation that's safe and effective. It's got value proposition for patients. And a culture of engagement, customer service, and collaboration. We're going to work with people, and that's industry, but other customer groups, patients, providers. We changed our programs, streamlined them, made them more predictable, and introduced new ones like breakthrough devices. And we advanced the science for supporting the design, development, and evaluation of technology, regulatory science, because a lot of innovations, the science isn't there to understand what they do. We really invested collaborating with others for building those kind of tools like bench testing, modeling, biomarkers, clinical outcome assessments, and made that available to innovators. And part in collaboration, and the last thing I'll mention, we helped co-found public-private partnership, now a 501c3 organization, the Medical Device Innovation Consortium, which is the only such entity here in the US devoted to advancing regulatory science for medtech. You don't know that group, they have a lot of great tools. Check them out, mdic.org. I do get part of their proceeds, so please.
Thank you, Jeff. Patrizia?
Very happy to chime in. What really resonated from what Jeff said is the concept of responsible innovation. I really like that. I came from FDA after spending time in academic medicine. I was also an investigator in clinical trials and then spent many years in industry. And I think it's really important that we make a differentiation between what drug developers do and what we do at FDA. We certainly have a very important role in regulating and to the extent that we can, obviously streamlining and helping drugs getting through development, but we're not drug developers. We're not collaborators of drug developers. We're fundamentally regulators. Now, we do have some very important tools that can facilitate drug development, that can sometimes expedite drug development, and if not, and tools that could actually potentially also slow it down, right? So it's really very important that we regulate drug development in an agile fashion, looking for ways to cut unnecessary sort of bureaucracy or hurdles and et cetera. But I think it's very important that we make that differentiation. And my many years in industry where I have seen certainly heroic efforts and very innovative efforts, but also development decisions that were largely driven by financial considerations. And we see a lot of Me Too's in drug development as well, which when we have many areas where there's a lot of unmet medical needs. I mean, rare diseases is one of them, but still lots of cancers that don't have treatments and so on. So this concept of responsible innovation from a regulatory standpoint, I think it's very important and fundamental. And I see that getting a little blurred these days.
All right, thank you. Thank you for the introductions and hearing about your background and your journeys coming to this point now. Patrizia, I'll stick with you with this next question. This is under the auspices of product approval considerations. What types of drugs are eligible to be considered for accelerated approval? And what distinguishes accelerated from traditional approvals?
Well, this is a big question, and I'm hoping that Dr. Marks will chime in at some point. So we can use accelerated approval in situations where we're dealing with a serious life-threatening disease that also does not have alternative treatments. Accelerated approval is possible if we can identify a surrogate endpoint, being usually a biomarker of the disease, or an intermediate clinical endpoint. And obviously, the most notable example is oncology, where they use a progression-free survival as an intermediate clinical endpoint to support accelerated approval. When we look at these endpoints in accelerated approval, we still apply the substantial evidence standard, as we do with the more proven clinical endpoints that we use for full approval. It is important to understand, however, that substantial evidence standard is applied in accelerated approval, is applied on endpoints that reasonably predict clinical benefit. And that's very different from having been confirmed to measure clinical benefit, and which is why we need to have confirmatory trials after we approve a drug with using accelerated approval. And these confirmatory trials are very important. It's important that they be adequate, well-controlled clinical trials, and it's important that they be conducted in a timely fashion. And unfortunately, we still see some sponsors who, I'm sorry, I can't say it any other way, that really drag their feet, that take years and years and years to do a clinical trial that, may take much less time. The other thing that is important to understand is that when we use accelerated approval, we accept a level of sort of residual risk that is much greater, or, greater, maybe much greater, but greater than what we accept when we use full approval. And that explains why sometimes the confirmatory trials don't confirm clinical benefit. And we have seen this in many areas. We have seen it in oncology. We have seen it in rare diseases. We have seen it in women's health, a very notable example last year, and so on. So hopefully I was able to sort of unpack something that is rather complicated and causes a lot of discussions even at FDA. And I'm going to turn it over to Dr. Marks to see if he has anything to add.
Yeah, no, I think the only thing I would add is that the last year we did get some additional authorities to help those essentially move along those who might be dragging their feet by being able to insist that people have some timeliness in how they go about getting confirmatory information for accelerated approval. It is this concept of residual uncertainty that needs to be resolved, and some people like having uncertainty. It's very good for the bottom line sometimes, and unfortunately, that's not good for public health. The faster we can resolve residual uncertainty, the better for public health. So I think the ability to move that along is probably an important piece of us being able to continue using the accelerated approval pathway to be able to hopefully get products to people in need faster. So I think hopefully people will take that seriously. We're taking it seriously. Also, I think the other piece, one last thing, is that the ability to have a more structured process for withdrawing products when they don't meet their their confirmatory endpoints. I think it's also helpful. It used to be a lot of appeals that would take a lot of time, and I think we now have a much more structured and hopefully faster process in making that happen.
And we have already used these two authorities once each, one that Dr. Marks knows very well. And we also recently issued a complete response to a company, to a sponsor who, when we determined that the confirmatory trial was not sufficiently underway. So.
Thank you. Thank you both for that. Jeff, we'll go to this next question with you. In April, so just about a month ago, you published, or the center published, a report on medical device innovation. Can you tell us a little bit more about this report and its intent and what you were looking to accomplish?
Yeah, certainly. So as I mentioned, we've been on a bit of a journey for the past 15 years. And, as Dr. Califf mentioned, last year we had that banner year of, 124 novel device authorizations. But 15 years ago, it was 25. So there's been a 5-fold increase, and that's been increasing over several years, kind of building up over time. And so we kind of thought it might be time to kind of tell the story a little bit. And that's what the report does. Very high level, lots of pictures. You can read it in 5 minutes if you haven't already. It's wonderful read to your children at bedtime. It goes very quickly. So it was an attempt to connect the dots, but also we highlighted some of the key actions we're taking in 2024. Some pertain to the TAP pilot and home as a healthcare hub, Dr. Califf mentioned, but another one is on a complete redesign of our pre-market program. We're really completely reimagining the enterprise with the goal of significantly reducing burden both to sponsors and to ourselves at the FDA, and make this a much more expeditious process, far beyond what our commitments are in the user fee program. And already we're setting up to do several pilots. Some are expansion of our triage program that we've done in IVDs, where kind of the lower risk 510Ks with a high quality submission, those will do a review in 30 days. One we did recently on a fentanyl test strip was in 16 days, to give you an idea. A second is to be able to do the review, if you will, in one seating. You can get up because it could take several days. We let people go to sleep, but it really is stay focused. And the third is on agile queuing. So things kind of are moving through, they're not sitting in the queue for someone to get to them. That's just a few things that we'll be piloting and a lot more that we'll be talking about later in the year.
Thank you, Jeff. And we have a printout of the innovation report. I have a copy of it at our booth outside, so check it out later today, as well as the CDRH statement on this report.
Elias will be autographing it.
I will be autographing it and auctioning it off to the highest bidder.
It will be worth something.
Well, I don't, maybe for my mom. Let's continue this discussion. When we're talking about innovation, obviously we're talking about also advances in technology. So Peter, I'll come to you for this next question. How would you describe the current landscape of first-generation transformative innovative products to reach the market?
Yeah, so maybe I'll take that as in terms of the gene therapies that we're seeing coming along. So the first generation of gene therapies that we're seeing, it's quite exciting because they're offering people a lot of promise in the rare disease field, and for some conditions that have had treatments, but now gene therapy potentially offers a much more convenient or a much fuller treatment. I think in some cases, it's really quite transformational. For instance, for patients with severe sickle cell disease, having the cell-based or these cell-based gene therapies, 2 of them now, one the CRISPR-Cas9 genome edited product that Dr. Califf mentioned in his talk. Those are really potentially major transformations, and although there probably will be better successor products to those, I think those may be around for a little while to come. On the other hand, there are probably some other first-generation products, like some of the products in the hemophilia space, where I suspect for hemophilia A, we'll see better products coming down the line that might have better durability, et cetera. I think we're still getting a sense of how these products work. The first generation of products that we're seeing are that are cell-based, or the ones that are being directly administered using the adeno-associated viral vectors are slightly problematic because you can't currently redose those products. So if they stop having effectiveness, you have to move to something else. That may change, and I think what may change, and it's just kind of over the horizon, is the use of CRISPR-Cas9 genome editing tools, which can be potentially administered using non-viral vectors such as lipid nanoparticles, something we've become very familiar with during the COVID-19 pandemic in the form of the mRNA vaccines. And so we may see second generations and then third generations of these gene therapies coming along in the next 5 to 10 years. So I think this first generation, extremely exciting. It's going to really improve a lot of people's lives right now, but I think more to come as we see the innovation build. And that's not uncommon. I mean, I think it happened in monoclonal antibodies. You know, the first generation monoclonal antibodies were a lot of these were murine antibodies that had their own issues of human anti-murine antibodies. It's all sorts of issues that came up, and then you know, the successive the development has really brought them forward. So I think that'll happen in gene therapy as well.
Yeah, there's so many examples, you know, elaborating on what Peter just said of the journey that, you know, where gene therapies are sort of embarking now. If you think about the HIV drugs, and what the first generation of these drugs were and where we are now. Think about Alzheimer's more recently. You know, you know, we have some, for the first time, some drugs that seem to have an impact on progression, but they also have, you know, some side effects and so on. And, you know, these, we can consider these, you know, these stages of development in major diseases as you know, a progression towards better and better drugs. We've seen it with cancer. Think about how toxic chemotherapies were only 20 years ago and where we are now, which is why it is important, despite the fact that some of our approvals or decisions may be viewed as controversial sometimes, to keep the field going, right? Because drugs get better over time, right? But if we don't allow the first, maybe not perfect, not entirely perfect wave of drugs to be tested and to be used, and for evidence around their use being generated, then we really, you know, cannot advance the field, right? So it is a difficult decision from a regulatory standpoint, because it comes down to accepting some residual uncertainty to Dr. Mark's point. So.
Absolutely, the benefit-risk calculation.
Exactly,
Absolutely.
and benefit-risk calculation. That's always overarching, right?
Yes. So. Thank you for these remarks. I think we're now ready to start to get some questions. So for those of you in the audience, I welcome you and invite you to come to a mic. Meanwhile, we'll start off with our online audience. And Larissa, I want to turn the floor over to you for our first question from online.
Yes, we have some. We have some good online questions here. The first question goes to Dr. Cavazzoni. What role does artificial intelligence play in safety assessments, in your opinion?
I couldn't hear the end of your question. My apologies.
What role does artificial intelligence play in safety assessments, in your opinion, from the perspective of the Center for Drugs?
I can tell you about, you know, today's role. I think the role will definitely expand, and you know, Dr. Shuren and Dr. Mark are really, I'm sure that will want to chime in on this. So to give you an example, we have been using, for now several years, we have been using machine learning to triage and deduplicate adverse events that are reported to MedWatch. That has been ongoing for for now several years. And we used to have a team of contractors who would do this manually. Having said so, I think that machine learning and artificial intelligence as applied to safety will be increasingly important in identifying trends in signals that may point to safety issues. I think that, there's a lot of discussions about how in silico data can be used in the evaluation of real world data and real world evidence. And I think that safety is probably one of the prime areas where we have used real world data and real world evidence. So there will be a lot of new applications. And I'm sure that we will be confronted with this very soon, if not now.
Thank you very much. Let's take our first in-person question.
Okay, great. The FDA Modernization Act talked about reducing animal testing. And I'm just wondering where you've seen innovations in that regard. Has there been a reduction in animal testing? And when you're talking about residual risk and potential, is it more in the pharmacology side? Is it more in the safety side? Where reduction in animal testing has been seen in drug development? It's an IND thing, so a lot of it isn't public. So I'm just wondering if there's some examples you could give on where animal testing has been reduced, and all three of you.
I can start that at least in the vaccine world, release testing has changed from certain tests used to require, for instance, non-human primates for release testing. Now that has transformed. I mean, they still some of these still use animals, but instead of using non-human primates, they can use rodents. In some cases, we're starting to see people looking at during developing gene therapies where they're using organs on a chip, because especially if you're developing a human genome editor, it's much more useful to have a human organ system such as has been developed at NCATS at NIH on a chip, and to look at that than to look at even a non-human primate, because the sequence isn't the same. So I think we're seeing a transition here. It's not night and day yet. And there still are things where we still need animals, where animal experiments are still bringing important information to the field. For instance, mouse experiments in COVID-19 vaccine development still are very useful. But I think there has been this shift from higher species, non-human primates, dogs, cats, to rodent models, and then from even some of the rodent work to in vitro or in silico work.
And I think all the centers are intentionally looking for ways to reduce animal testing.
The 3 R's, right, and to the extent possible. And for instance, non-human primates, there is a program that they're either reused or rehomed at this point.
And the science needs to get there, right? So we'll be able to reduce animal testing even more as the science gets there. But the reality is that today we still need to do animal toxicology studies in many instances before putting a drug for the first time in humans. Certainly the organ on the chip technology is advancing. It is not there yet when we're talking about complex multiple complex physiological systems that are sort of interacting. But, the science will continue to advance.
I think too, we're also looking at how we reduce testing on people as well. So we've got, in silico clinical trials, phantoms used, with radiological imaging. If folks are interested, certainly on the medtech side, we have some of those tools available, regulatory science tools. We've got a catalog up on our website. So it is something to check out. There may be something up there that's relevant for your technology.
Thank you. Thank you for the question, and thank you for the responses, my panel. I'll take a question from this microphone.
Thank you. This is directed at Dr. Shuren. So Greg Cornever, Avanos Medical. It's been my experience that that there's this reluctance among the decision makers in medical device management level people to avoid a de novo at all costs. And PMA 510K, but de novo seems to be this fear of the de novo. Can you address that and assuage that kind of fear?
Yeah, I don't, so I've not experienced the fear of a de novo. Years ago, there was the issue around that because the de novo program was broken. It was really hard. We needed changes in the law. So we used to try to shoehorn technologies that really were for de novo into 510K because the de novo path was just terrible. So we actually have the highest numbers of de novos coming through in the past few years than we've had, for previously for the history of the program. So they are coming through. I think there's a different issue with de novo, is that there's more of them that don't get authorized than what we see for the rates for PMA and for 510K. And we've been looking into it a little bit, and a number of times these are issues around, also because it's so new. Sometimes the developers too don't seek out advice, and they come in the door, or they're thinking they're coming in one pathway and haven't had the conversations with us. So I would say, if you're looking at de novo, one piece of advice is make sure to come talk. We provide lots of opportunity to get on the same page. That's the most important thing to do.
Good question. Larissa, I think I want to go back to you for another online question for our panel.
Sure. Dr. Shuren, last month, CDRH launched the Home as a Healthcare Hub initiative, which is aimed to help home environment as an integral part in this program, and how it may impact the advancement of innovation.
This is part of our strategic priority on advancing health equity. I think, as everyone knows, there are millions of Americans live in primary care deserts. And in part, one of the challenges, we built our healthcare facilities administrators. It's not designed to meet the needs of patients. I'm a patient myself. I think the healthcare system sucks, quite frankly. Drive down costs. A lot of costs come out of dealing with things through brick and mortar facilities. Then we have to rethink how we deliver care and design it around people and bring it to people rather than expecting kind of where they are, like the home setting writ large has to be designed for it. And technology is a bridge. It's not just to connect a person with their provider, to provide interventions and disease management. You've got to design it for people. So this initiative on Home as a Healthcare Hub is to do just that. We've partnered with it to build a virtual reality prototype. And we're doing this collaboratively with, for underrepresented populations in mind, and to start with diabetes. So we're partnering groups, provider groups, medtech, innovators. And that prototype is designed, will be an idea lab to help innovators then kind of think about how they would design technology, because we really need not what happens today, a lift and shift from the hospital to the home, which is, and then jerry-rigging it. It doesn't work that well. It's a Band-Aid, and it's what you do when you got nothing else. But also, no one wants the hospital in their home, quite frankly. I don't want a nurse waking me up every 2 hours, and I'm not eating green Jell-O. It's out of the question. But we need integrated technology. We need it that's medical grade, but a consumer kind of product. That's for people to use. So that's what that prototype will help too. And our goal is to have that kind of launched and out there, at least a prototype later this year.
Thank you, Jeff. Thank you for the question. Let's go back to this mic for a question from our audience.
Thank you so much for this opportunity. So this last decade was an innovation for oncology, and I think we are entering the decade of innovation for neurodegenerative diseases. You touched upon Alzheimer's. I wanted to get your thoughts on Parkinson's disease in particular, because biomarkers are still being researched, and then digital tools and wearables and endpoints. So I think this question is for all three of you. What do you see innovation in Parkinson's disease in the future?
I'm happy to start us from sort of talking about the drug side. Parkinson's disease is another area where there's a lot going on, beginning with moving from a diagnostic construct based on symptoms to a diagnostic construct, as I'm sure you know, based on actual sort of biology, biomarkers, and so on. And it's the same shift that we have seen with Alzheimer's. And so once that shift is made in how a disease is diagnosed and characterized, then it opens tremendous opportunities for drug development, right? Because then it is possible to use biomarkers in drug development. It is possible to develop drugs that have more precise or targeted mechanism of action, and so on. And so what's going on in Parkinson is very exciting, and it goes sort of hand in hand with what we have seen with Alzheimer's, and what we hope that we'll be seeing more and more in other neurodegenerative diseases as well.
I don't have a whole lot to add except to say that we are starting to see cellular therapies where people are using induced pluripotent stem cells, trying to deal with trying to address this, but they rely a lot then going back to what Patrizia was talking about, assessment methods to try to see whether they're working or not.
And I think complementary to that, also looking at other kinds of measures in partnership with others like Michael J. Fox Foundation that can be used in the home setting. So we can also increasingly move towards decentralized clinical trials and broaden participation. That's critical. And then, of course, MedTech has its role. There are, stimulation devices that we have today, but also thinking about the roles with other kinds of technologies and AI that can be assistive in Parkinson's disease patients, because certainly things around gait visual cueing plays an important role, and technology is a way to help provide that to folks. So there's a lot of innovations underway, and all of this is, and I think the exciting part is not to think in individual spaces, but thinking holistically for people, and kind of that combination in the drug, biologic, medtech space, and provide holistic solutions for people with Parkinson's and other neurodegenerative diseases.
Thank you, Jeff. We're very rapidly running low on time. This has been great. We could probably spend 3 hours talking, and it'd be so much fun. So we'll try to get through as many questions as we can in the next 5 minutes we have left. Larissa, I want to turn back to you for any questions from our online audience.
Yes, there is another question, and this question goes to Dr. Marks. So someone says, I heard you recently said that we need a $4,000 kind of computers before we get to $300 computers, and that was a metaphorical expression that was said in the context of the development of cell-based therapies. Can you please talk a little about this and tell how this metaphorical expression relates to innovation in the field of cell and gene therapy?
Yeah, so in the area of cell and gene therapy, and it could be either, but we've started with a first generation of products where the first generation of chimeric antigen receptor T cell therapies were about $400,000 to $500,000 for the product, let alone all of the other care that went around them. And I think one could say, well, that's crazy. It's an incredibly high price, but that was what it cost to get that innovation out there. But what we're starting to see now is the successive rounds of innovation, where now the innovation is in how can you make those products more accessible by bringing their price down by an order of magnitude. And probably within the next few years, we'll see chimeric antigen receptor T cells out there for a tenth of that cost. Either because of decentralized manufacturing efforts, or because some manufacturers, that's precisely what they're trying to do, is develop a product in a much more efficient manner for less cost. So I think, like many things, as we have more use, more development, we will see these issues come down. Part of it too is that the people making them initially have had to make them by hand largely, and then as the market has expanded, we're seeing fabricators who make devices to manufacture products making systems such that we actually we have like closed system devices that can make these products, again bringing down costs. So I think that was what that comment was meant to explain.
Thank you, Peter. Let's take a question over here.
Thank you for taking my question. Actually, it's to Dr. Marks as well. I mean, I heard you, and I fully agree with you that what we are seeing for the gene editing aspects is the growth pains that we got through the ABs. But I have a specific question. I was able to see the advisory committee for the XSL product and it was quite interesting that out of the questions that are being asked by CBER, there's a need for standardization, meaning that I would like you to elaborate a little bit more on what will be the agency's future in terms of defining risk assessments aspects on gene editing risk, of errors. So please elaborate on that.
If I understand the question correctly, it's how do we deal with the risk-benefit assessment here in a more standardized manner?
Sure, and particularly that aspect of gene editing errors.
Yeah. So right, outstanding question. So the issue for genome editing is this concern about off-target effects. There again, some of that may have to do with the generations of genome editors. The first generation genome editor, CRISPR-Cas9 constructs, we've seen the ex vivo approach was very helpful because you could sequence afterwards and figure out what had happened. You could use in silico analysis to look for off-target effects, and you could use the cell-based analysis. And so right now for these first generation editors, we're using essentially two different methods, at least, or asking people to use an in silico method and a cell-based method to try to de-risk things. I think ultimately what will de-risk them further is moving to new generation CRISPR constructs, for instance, CRISPR base editors and CRISPR prime editors. The construct itself doesn't cause a double-stranded break like CRISPR-Cas9 does. Double-stranded break's bad because you've got DNA flopping in the breeze. Base editors and prime editors. Prime editors are basically a CRISPR construct which has been grafted to a reverse transcriptase, so it can actually essentially read-write essentially from the RNA template. It doesn't cause a double-strand break. Again, less potential problems. Still, you have to worry about off-target effects. Idea here is to look by essentially two methods to try to minimize off-target effects. And to date, at least the data coming out of some of the non-human primate experiments, as well as some of the initial, very initial experiments with initial CRISPR directly administered to humans, is at least looking encouraging.
Thank you, Peter. Thank you for the last question. And I think that will conclude our session for today. I want to thank our panelists for joining us on this great topic. Again, we could talk for hours on end. Thank you for interacting with us both online and in person. Let's give a final round of applause for our